All right, the last class of the major molecules of life that we need to get into are going to be the nucleic acids. Each cell stores information for how the cell should behave and respond to the environment, how to replicate. Everything that that cell needs to know about being a cell is encoded for in its DNA. So this information is stored in the form of nucleic acids. Nucleic acids are polymers of nucleotides. So just as an example, some of the nucleic acids that we use for storing information would be deoxyribonucleic acid, or DNA, or ribonucleic acid, RNA. Those are going to be the two main types of nucleic acids that we are going to be discussing in this course. DNA and RNA have structural differences related to the sugars, the bases, the number of strands, and we'll get into great detail about those differences as it comes up. Both, though, are going to be polymers of smaller subunits called nucleotides. A nucleotide is going to be made up of a 5-carbon sugar that's bonded to one of 5 nitrogen-containing bases in a phosphate group. What we have here is a nucleotide. This nucleotide shows us, this shows us the basic structure of the nucleotide, the phosphate, the pentose sugar, and then the nitrogen-containing base. This region over here, the nitrogen-containing base, it's our R group, our variable group, much like amino acid sequences where the bulk of the amino acid is going to be the same, but we have one R variable group. Nucleotides are going to function the same way. Each is going to have the same phosphate group, the same pentose sugar, and then a unique or separate individual nitrogen-containing bases. The sequence of nitrogen-containing bases right here is going to be what gives us our DNA sequence. So shown here, we have a section of DNA. Sample of DNA. DNA is one type of nucleic acid. It's going to be storing information for replication and protein synthesis. DNA is what we use ultimately for housing and storing that information. RNA, ribonucleic acid, is going to be a little bit different. It doesn't store information, but instead uses that information stored in DNA. This is going to be used for protein synthesis and regulation of gene expression. You can kind of think of DNA and RNA as tools that you would use for this class. So for example, DNA is going to be your hard copy textbook. Everything that is in that textbook is of importance, but not everything in it is of importance every moment, all the time. When you are studying for a particular test, do you go to your notebook or do you go to your textbook and copy down every single word that is in that textbook? No. At least I hope not. If you are studying for this particular chapter, I hope that you are not taking the time to copy down word for word every single word in your entire textbook. That would be insane. What do you do instead? Instead, you're going to read the material, copy down all of the relevant information into your notes. You're then going to take those notes, study, utilize them, and turn them into a project, a homework assignment, a test. RNA and DNA work the same way. DNA houses all of your information. It is the hard copy. We don't want to touch it. We don't want to ruin it. We don't want to lose a word of it. Every bit of it is important. So we're not going to damage it in any way, but we're going to leave it stored up inside the nucleus where it's going to be nice and safe. When we want to make a protein, we're not going to copy down the entire sequence of DNA. That's insane. We don't need all of the DNA. We are only going to need a small part. So if this part right here is going to code for the protein we want, we're going to use RNA. We're going to copy down this message right here. We're going to leave the nucleus. We're going to go make ourselves a protein just using that little bit of information right there. So DNA is the hard copy complete textbook. RNA is going to be your quick notes of just the important information you need right now in this moment. Another form of nucleic acids are going to be coenzymes. We've already mentioned that cofactors can be things like vitamins and minerals to help enzymes along. There are also coenzymes that are made up of nucleic acids. These are going to be used to help regulate enzymatic reactions. We're also going to find that a very important nucleic acid is ATP, adenosine triphosphate. ATP is going to be instantaneous energy storage. It says energy storage, but really that's a bit misleading. You can kind of think of carbohydrates as short-term energy. short-term energy and lipids as long-term energy storage. ATP is instantaneous energy. For me personally, just as a anecdote, I like to think of this in terms of money. For me, ATP is cash. As soon as I have cash, I spend it. I can't hold on to cash for anything. When I got my very first job, I also got a debit card and direct deposit. So for me, a credit card, a debit card, that is hard-earned money. I do not spend that for anything. But cash is birthday money. Cash is gift money. Cash is money that is meant to be spent. I can't hold on to cash. If I have it, it's gone, it's spent. That's ATP. As soon as you have it, it's gone, it's spent. If I ever need cash for anything, I withdraw however much I need, and I spend it right away. I don't carry cash. I don't store money in the form of cash. So ATP is cash. Carbohydrates is the checking account. The money is there. It's going to stay there temporarily. You're going to put money in, you're going to take it out. So carbohydrates you're going to build them up to store energy temporarily. You're going to break them down to store energy temporarily. Lipids are your savings account. You're not going to touch it unless you really need it. So for me, that's how I like to think about it in terms of energy storage. ATP, instantaneous. As soon as you have it, it's gone. And same with your carbohydrates. Your carbohydrates are going to be your checking account. It's there. It goes, flows in, flows out, flows in, flows out, can stay there for a while, will eventually get used. Lipids are going to be sort of your long-term savings plan. You're not really going to touch it unless you absolutely need to. All right, so those are our nucleic acids. Table 2.4 is an excellent source to help you sort of review the characteristics of DNA and RNA, some of the similarities, some of the differences. We are going to spend a lot more time on the similarities and differences at a later date. We'll touch on it briefly, but Table 2.4 is a great summary of the differences between DNA and RNA. We're going to touch on some of those differences just briefly. DNA, like we've said before, is going to be used for storing genetic information in your cells. It's ultimately going to store them in a structure known as a chromosome. DNA is going to be made up of twisted double strands. DNA is double-stranded. We have one strand here, and then we have an anti-parallel strand running the opposite direction. This is what's known as a double helix. These bonds are going to be held together by these dotted red lines. Does anybody remember what those dotted red lines symbolize from earlier in this chapter? Excellent! Those are hydrogen bonds. Those double helix is going to be held together by hydrogen bonds that are formed in between these nitrogenous bases. There are going to be 4 nitrogenous bases found inside of DNA. They're going to be cytosine, thymine, guanine, and adenine. They're abbreviated by one letter, ACTG. Adenine, cytosine, thymine, and guanine. So those are going to be our 4 bases for DNA. Ribonucleic acid, RNA, is going to be ever so slightly different. As we've already mentioned, its job is not to store information, but instead to convert and utilize that information. You're not going to store your information long-term in the form of notes. Information is stored long-term in the form of textbooks. However, you are going to use your notes to convert that information from DNA into proteins, or from hard copy textbook into usefulness. So we're going to convert the genetic information in DNA into proteins. It is single-stranded. We do not have a double strand over here. We don't see any hydrogen bonds going off to another strand. And it's going to have 4 nitrogenous bases as well, cytosine, guanine, adenine, and uracil. Uracil is going to be the difference here. We no longer have thymine, but uracil replaces thymine. So that's our DNA, our RNA. Next, we're going to talk about ATP, adenosine triphosphate. This is the nucleic acid that is actually used for energy storage, instantaneous energy storage, very, very short energy storage. ATP, adenosine triphosphate, is a nucleotide that's composed of an adenine, a ribose sugar. Adenosine is that ribose sugar, and 3 phosphate groups. So that's where the name comes from. Adenosine, the adenine, the ribose is adenosine, and the triphosphate, the 3 phosphate groups. Here's what that looks like. Here is going to be our adenine. Here is our ribose sugar and the 1, 2, 3 phosphate groups. Now this is going to be a very high energy molecule. The reason that we use it, this for energy storage is because we can store a ton, crap ton of energy right here in this final bond between these, the second and the third phosphate group. The hydrolysis of this bond is going to yield the molecule ADP, or adenosine diphosphate, an inorganic phosphate, and a ton of energy all at once. This energy can be coupled with chemically unfavorable reactions to allow the cell, to allow reactions to proceed. So just for example, key steps in the synthesis of carbohydrates and proteins and muscle contraction and nerve impulse conduction, those are all unfavorable reactions. But we add a burst of energy and boom, we can drive those reactions forward. So ATP is called the energy currency of the cell. The more ATP you have, the more metabolic reactions you can drive forward. The more carbohydrates you can make out of, the more polymers you can make out of monomers with the more ATP that you have. So this high energy molecule, all of that energy is stored in this final bond right here that is very unstable. So here's what happens when we have our ATP. When we break our ATP, we're going to release, boom, huge burst of energy. We're going to make adenosine diphosphate, ADP. We go from triphosphate 3 to diphosphate 2. And we're going to have an extra phosphate group just hanging off because we snapped him off, and a huge burst of energy. Interestingly enough, anytime we want more ATP, we can make it. We can add energy plus a phosphate group and an ADP, and we're going to make ATP. So anytime we want cash, we can make it. Anytime we need to spend cash, we can. ATP is going to be instantaneous. We can build it up, we can break it down whenever we need to. But in order to build it up, we need to have ADP, phosphate, and energy. We need to put that work in, just like, it takes energy to make cash, it takes energy to make money, it takes energy to produce that ATP. So there you have it, ATP. All right, and that's it for this chapter. It's a very big chapter. I think this is going to be the largest chapter that we study this semester. Typically, we spend 2 weeks on this chapter alone, but because we're on accelerated course for the summer, we took a little bit more time with it. So, at the end of this chapter, you should be able to describe the characteristics of the subatomic particles, protons, neutrons, and electrons, meaning I want you to know their location, mass, and charge. You should also be able to explain the structure of an isotope. How is an isotope different from a regular atom? You should also be able to differentiate between a covalent, ionic, and hydrogen bonds. You should know how to differentiate them in terms of strength and also the actions of the electrons, meaning how are those bonds formed? How is a covalent bond different from an ionic bond? How is a polar covalent bond different from a non-polar covalent bond? You should be able to explain all of those. You should also be able to list the unique properties of water that make it valuable to the biological systems. Not just list them, but understand them. Understand where they come from. How does hydrogen bonding play a role in all of these properties? How does the polar nature of water play a role in all of these properties? You should also be able to predict what happens when you add an acid or base to water. You should be able to define pH, explain the range of the pH scale, and tell which values indicate acid and which values indicate a base. What is neutral pH? What's an acidic pH? What's a basic pH? You should also be able to describe the structure of a polymer, including its formation through dehydration synthesis and the breakdown through hydrolysis. You should be able to describe the structure and biological purpose of carbohydrates, lipids, proteins, nucleotides, and also give an example of each. And then finally, be able to describe ATP as the currency of the cell. How do we get energy from ATP? How do we put energy into ATP? And that is it for this chapter. Please let me know if you have any questions or if there's anything that you would like me to explain slower or more detail. I am happy to make a second video going over any topic that you would like to discuss from this chapter. So please just let me know.